For the second part of the experiment, we will be exploring the effects of activators and inhibitors on the Krebs cycle. To begin, 520 ml test tubes were used to prepare 5 different solutions A to E. To each test tube, 50 microliter of 0.1% methylene blue was added and afterwards shaken to mix contents. The test tubes were then tilted in order to add 500 microliters of mineral oil. Afterwards, the test tubes were incubated in a 37 degrees Celsius water bath for about 2 hours. Before the Krebs cycle can occur, oxidation occurs to convert pyruvate to acetyl-CoA, which is considered the link between glycolysis and the Krebs cycle. The main function of the Krebs cycle is to provide reducing equivalents through the oxidative decarboxylation of acetyl-CoA, as well as providing intermediates for biosynthetic reactions. For the experiment, methylene blue is used as an artificial electron acceptor instead of using NAD+ and FAD. As an indicator, methylene blue is blue in its oxidized state. On the other hand, methylene blue becomes clear once reduced. This property is useful for observing the metabolic processes happening in yeast, as the disappearance of the blue color indicates the rate of yeast metabolism. Since the experiment relies on the ability of methylene blue as an artificial electron acceptor, it is important that hexane or mineral oil is added to stop oxygen from entering the solutions. This prevents the oxidation of methylene blue, which may cause false positive results, as well as prevent the production of parasitotoxic reactive species, particularly hydrogen peroxide. As seen in the slide, the figure on the left shows the test tubes before incubation, while the figure on the right shows the resulting change in color intensities after incubation. Test tube C is observed to have the greatest change in color intensity, followed by test tube E, B, then D. On the other hand, no observable changes were observed for test tube A. Initially, test tube A has the darkest blue color, which may be due to the amount of water present in the solution. This is due to a small amount of oxygen that shifts the equilibrium of methylene blue to the left, forming a solution of higher color intensity. Recall that for test tube A, no color change was observed after incubation. This result is expected since the solution only contained water, which does not reduce methylene blue. On the other hand, white precipitate was observed for test tubes B, C, D, and E, which is caused by the settling of undissolved yeast during incubation. Comparing test tube B and C, it can be observed that test tube C has a lighter color due to the metabolism of glucose that would cause methylene blue to be reduced, consequently lowering color intensity. For test tube D, malic acid was added in addition to yeast and glucose. The ionized form of malate is one of the intermediates of the Krebs cycle, subsequently oxidized to oxaloacetate by the action of malate dehydrogenase. Based on the Le Chatelier's principle, increased malate concentration shifts the reaction forward, reducing the NAD+ to form oxaloacetate. As a result, test tube D is expected to have a higher rate of decrease in color intensity. However, contradictory to theoretical results, it was observed that there was still little color change in the color intensity. This error may be caused by an insufficient amount of mineral oil that prevents oxygen to oxidize methylene blue. For test tube E, hydrogen peroxide was added in addition to yeast and glucose. This compound is responsible for limiting the amount of NADH by inducing oxidative stress. By inhibiting the activity of alpha-ketoglutarate dehydrogenase, hydrogen peroxide disrupts the Krebs cycle by limiting the amount of succinyl-CoA. Consequently, the amount of succinate is decreased and the activity of succinate dehydrogenase is reduced. As a result, no color change should be observed. However, it was observed that the test tube E turned into a lighter blue color, which indicates that the metabolic processes are not completely stopped. Conclusion and recommendation. To conclude, the effect of inhibitors and carbohydrate source on glycolysis may be determined by measuring the rate of carbon dioxide production in the processes that follows glycolysis, alcohol fermentation for anaerobic conditions, and the oxidation of pyruvate and the Krebs cycle for aerobic conditions. In addition, arabinose, sodium fluoride, and sodium citrate are inhibitors that lead to the decrease of the rates of carbon dioxide production, with sodium fluoride being the best inhibitor among the three, followed by arabinose and sodium citrate. Third, sodium fluoride completely inhibits carbon dioxide production when the substrate is glucose, but allows for minimal carbon dioxide production with fructose. Lastly, malic acid serves as an activator and hydrogen peroxide serves as an inhibitor in the Krebs cycle. It is therefore recommended that other monosaccharides like galactose and mannose and disaccharides like maltose, lactose, and sucrose may also be explored, and the effects of activators like insulin on glycolysis may be investigated. Here is a list of our references. Thank you.